Hello everyone. Today I'm going to be talking about liver anatomy and histology. I used two main sources here. Both of these are histology books, and I give you the page numbers in case you want to check in with something or follow along. There's a lot of really good high quality pictures in both of the books. And in addition, in the box folder I put a quick study sheet. It's about a page, and the PowerPoint presentation if you wanted to take notes on it. So let's review our actual anatomy of the liver. So if you remember, we're going to have portal blood flow into the liver from three different veins actually. Do you remember what they are? The splenic, superior and inferior mesenteric. And all three of those are going to be feeding into the portal vein here. So remember, this is quote-unquote dirty. It's come from the intestinal tract, and a lot of it is mostly deoxygenated, and it is coming back to the liver to get filtered. In addition to the portal vein, we're going to be having, so here's the portal vein, and then in addition we're going to have the right and left hepatic arteries. Those are from the heart, so those are fully oxygenated. And then going in the opposite direction is the right and left hepatic bile ducts. Together, these three structures, the bile ducts, the hepatic arteries, and the portal veins, create what is called the portal triad. And that starts right here, but it continues throughout the liver. You can see that each one of these branches has all three colors, kind of like Aquafresh. So like right here or right here, you'll see that every strand has basically the bile duct, some sort of an artery branch from the hepatic artery, and a branch from the portal vein. So once all of the blood from the portal vein and the hepatic artery is delivered to the functional unit of the liver, which is the hepatocyte, it basically gets filtered by the hepatocyte and then drawn out into these different veins called the central vein system, and goes out the hepatic vein and into the inferior vena cava. The blood flow here is, as I've discussed, the portal vein and the hepatic artery enter and they go into the portal triads. They're filtered through the liver lobules, and then they enter what are called central veins, and then sublobar veins, collecting veins, and hepatic vein. So these three here are not shown in this picture. They just made it a simplification by saying the central vein system, and I don't think we need to know that level of detail. So when I'm going to go into the more micro level of what a liver lobule looks like, but I think it's helpful to remember the main functions of the liver before we jump in there. One is to secrete and produce proteins, and remember, proteins are pretty large compared to normal molecules. Metabolize large molecules such as drugs, filter blood, and produce bile. So let's look at what a hepatic unit of operation looks like. So here are the hepatic units called hepatic lobules, and classically described they would be a hexagon here. So you can see a hexagon, and this is a hexagon with a bite taken out of it. And here we can see the portal triad. So I know that this figure has a lot of stuff going on in it, but just bear with me and just kind of follow what I'm talking about. So we have the portal triad here. So we have deoxygenated blood coming in through here and oxygenated blood coming in through here. They're mixing and heading and being filtered. So this whole section, as they're moving here, they're getting filtered and even more deoxygenated, and then going into the central vein. And then the central vein is going to eventually lead out to the hepatic veins that go back to the heart. And one of the main functions, one of the main ways that they are filtered is that they don't just kind of flow along the hepatocytes, they have these fissures. So these fissures are called sinusoids. So right here This is a sinusoid, this is a sinusoid, this is a sinusoid. And the sinusoids separate each one of these, what are called hepatic plates, these rows of hepatocytes. And as the blood is flowing along the sinusoid, it's able to exchange nutrients and get albumin and get detoxified with the hepatocytes. Now, which way is bile flowing? Well, bile flows the opposite direction of blood. This is very important. So bile is going to be created by all the hepatocytes, but then flow in the opposite direction. It's going to be collected in these things called bile canaliculi, and then put into one of the branches of the bile duct and head down out to the gallbladder. So now that we have a basic understanding of what a hepatic lobule looks like, unfortunately, the way that histologists decided to describe hepatic lobules is confusing. And there are actually 3 different ways to describe hepatic lobules. And I'm not really sure where these different distinctions come from, but I think that it's a good idea that we learn a little bit about each of them. So the first way is what's called the classic way, and that's what I've already kind of described, which is that there's a hexagonal shape, which in the middle has the central vein, and then there's 6 sides, and at each point is a portal triad. So we're missing one here, but you know what I mean. So this is the classic way to describe. So there would be a whole bunch of hepatocytes in the center here that would be filtering the blood and adding albumin and stuff. The second way to look at it is what's called portal lobules. And I don't understand the name distinctions, why they chose portal lobules versus why they didn't call it bile lobules, but basically it's the area drained by a bile duct. So if we look at one bile duct, say the bile duct here, and then we look at the area that drains to it, then we could draw a triangle here, and we could say that this was a portal lobule defined by that portal triad. The next way to define something is with the portal asinus. And a portal asinus is the area supplied blood by 2 hepatic artery branches. So if we look at 2 hepatic artery branches, such as here and here, if we look at the area that they supply, it would look something like this, a diagonal area. And one thing histologists like to do is break up this area into segments. So this first segment here would be zone 1, that's the most oxygenated, zone 2, the moderately oxygenated, and zone 3, the least oxygenated, as blood is flowing this way and being used by the hepatocytes. Here's a better illustration of what these mean. I think that this is, somewhat confusing, so I think this is a really good explanation, but just had kind of a lot of information on the page. But basically, we can see here that this is the classic deal, 6-sided lobule with the central hepatic venule in the middle, and all of the blood is going to be drained to the middle here. We could look at a portal lobule. This green is the bile. The bile is being drained into that bile duct right there. Or we could look at the portal asinus, in that we have zone 1, zone 2, and zone 3. So now I wanted to take a closer look at what's going on and actually look at some histological preparations. First I wanted to look at the flow of blood. So what do you think we're looking at here? Well, this is the portal triad. Here we have the portal vein. So this is bringing dirty blood from the mesenteric arteries and splenic artery. This is the hepatic artery, so that's bringing fresh deoxygenated blood. And this is the bile duct, so that bile is draining out. So the portal vein is draining into the sinusoids, and it's trying to reach the central vein. So if we assume that that there's a giant hexagon here, and that this is one of the corners of the hexagon. So we're going to be wanting to drain blood this way, and we could be following these sinusoids here. So these white things here are all kind of sinusoids. You can see some over here too. They're a little snake-like. Conversely, we can see here, the central vein. And so we're not able to see a portal triad here. So this is a central vein. So blood is going to be moving from portal triads to this central vein, like this. And you can see all of the sinusoids that it would travel on. Right here, right here, right here. And one thing to note is that the central vein doesn't really have a thick vascular wall. So it's pretty plain right here, very light. As opposed to, if we go back here and look at, so in comparison, the portal vein does have a bit of a endothelial wall, and here the bile duct definitely has a thick vascular wall. So that's a great way to be able to tell the difference between portal triads and the central vein, which we'll do later in this presentation. So what exactly is in a sinusoid? This is a very important part of the whole of all of liver physiology, since this is the primary area in which nutrients are exchanged. So it's covered with a fenestrated endothelial capillary wall, which means that basically it's just a endothelial layer that's fenestrated, just means very holy, and so this is like full of holes. And then between this endothelium and the actual hepatocyte, there is a perisinusoidal space, also called the space of Dease, where the microvilli of the hepatocytes can exchange their large molecules with those in the blood. In addition, there are two other types of cells, Kupffer cells that are macrophages that break down old RBCs, and Ito cells that store fat and vitamin A, and they also have a role in immunity. So let's take a look at this diagram. I really like this diagram, and I think it's a great way to study hepatic physiology or liver physiology. So here we have the portal triad. We've got the bile ductule, the portal venule, and the hepatic arteriole. And here we can see the the oxygenated blood mixing with the deoxygenated blood, and then headed toward the central vein, which will eventually drain into the hepatic vein. We can also see the Kupffer cells. Remember, these are the macrophages that work on cleaning the blood and removing old red blood cells. And we have a stellate cell, which is another name for the Ito cells. These are the fat-storing cells. These are important because they are going to be what proliferate in alcoholic fatty liver disease. Also, we can see the space of Dease, or the perisinusoidal space, and that's this kind of pink space in between the endothelial cells and the hepatocytes. And yeah, great. So let's take a look at what these look like IRL. So here is a scanning electron micrograph, and you can see here all of these fenestrations, these huge holes in the endothelial capillary wall. These are important because they allow all of the exchange to happen. In addition, another thing that helps the exchange happen is these microvilli. And so here is the perisinusoidal space, is what this arrow is pointing towards, and that's where a lot of the exchange happens. So let's say, you know, a big drug can go in here and enter one of these little holes, enter the perisinusoidal space, and then get sucked in by the microvilli of the hepatocyte. And then over here we can see the nucleus of the endothelial cell. We can see another hepatocyte. We can see more microvilli in the perisinusoidal space. Really cool. Here's another light microscope view. So here's the hepatocyte, perisinusoidal space, endothelial cell. Here is the actual sinusoid. Oh, it's So I guess it's this and two. This is the macrophage, and then these little globules right here are the fat cells. So I think this is more of a cross-sectional view, but I'm not 100% sure. So now let's look at the flow of bile. So as I said, bile flows in the opposite direction from blood, and it doesn't use the sinusoids. Instead, it uses something called the bile canaliculi. So in between each hepatocyte is a space where the bile collects, called the bile canaliculi. These canaliculi collect into bile ductules covered with the distinctive cuboidal epithelial cells. So here we can see a cross-sectional view, and we can see the hepatocytes, and then they are exporting their bile into these canaliculi, which will flow into the ductules. You can also note these distinctive, very vascularized cuboidal epithelium. Here we have another view of the hepatocyte, and we have the bile canaliculi right here. These areas here, I think, are supposed to represent the bile canaliculi, whereas this would be where the blood would be. So blood in the terminal end of the ileum. Remember, we're going to have bile salt absorption here, and 90% of the bile will be recirculated. 10% of the bile that we use is resynthesized, if you remember, synthesized from cholesterol, which turns into cholic acid, which is then conjugated with either glycine or taurine, and then exported. Another great illustration. And then here is the same illustration that we saw earlier, and we have the bile ductule, and you can see it is flowing in the opposite direction, going towards the bile ductule. And then we've also seen this as well, and I just wanted to show you it again to point out the great endothelial, or the cuboidal epithelium of the bile ductule. Here's another really great view. So this is of a hepatocyte, and here we see the sinusoid. We see the perisinusoidal space. We see an epithelial cell, and here's the bile canaliculi. So bile is going to be exported in from the hepatocytes in here. Here's another view. Here's the bile canaliculi. This is a really cool view. So here's the hepatocyte, and they're just going to be exporting it into these canals. Just amazing that they're able to get these photos. Great. Now here's an unlabeled slide, and can you figure out what's going on? So this is the portal triad here. So if we were going to look at a classic lobule model, then this would be the corner of the hexagon. So blood is going to be flowing. I think that this, because it doesn't have an extreme amount of vascularization, is going to be the portal vein. So blood is going to be flowing out here through the sinusoids and into this central vein. And then I'm not sure which one of these is the bile duct. I think it's this one, but I'm not 100% sure. And I don't think we really need to know, as long as we're able to kind of identify where the portal triad is, and it's pretty clear. And to kind of wrap things up, I just wanted to show this picture because I think it's interesting. Here we're missing, you notice this area, this area here doesn't have very much vascularization, so I think this is the the central vein, central vein. And then I think this is the portal triad. And I think this is probably the portal vein, and I can't see the bile duct. But what looks different about this? Well, we have these huge deposits here. So this is alcoholic fatty liver disease. And these are the Edo cells storing a whole bunch of fat, and obviously this liver is not going to be able to move blood from here effectively through all of these fat nodules to get it to the central vein. So this is a good view of pathology. I think we could probably do another LO on pathology. All right, thanks a bunch. I will see you in class.